to look at it is through a gray to green kind of continuum of approaches. So the gray and the green refer to the colors of these different stormwater control measures. They're also sometimes called BMPs, somewhat related terms. And the gray, an example of gray infrastructure is just like concrete pipes or underground concrete basins. Things perhaps like underground injection control devices or dry wells. And then there's sort of like a lot of steps in between, such as like infiltration basins, maybe planter boxes or rain gardens on the sides of roads. And then on the far green side are fully untouched natural areas that just do what they do to treat stormwater just by existing and being healthy and functioning. So where this constructed wetland I studied falls along the spectrum is as a kind of a stormwater pond and wetland. It's not entirely natural, it's man-made, but the way it works is based on the functioning plants and processes that occur within it. So what are constructed wetlands? I'm going to use that term a lot. Like I said, they're man-made, human-made, sunlight-powered mimics. They don't have pumps or use electricity. They just are there growing plants and providing these ecosystem services I list here on the left. A lot of them some of them are more important to certain managers than others, like improving water and air quality or reducing peak runoff. There's also nice services they provide, such as educational, which is kind of what's happening right now, where we can study these things and learn about them. Also recreational services, such as walking around these systems and seeing, all sorts of beautiful birds. Definitely not a bird enthusiast, but it was really cool to work in the wetland and see the different birds that were flying around. And how they work, I won't list these processes off, but there's a range of them, ranging, starting with biological or animals degrading solids in the water column, or plant uptake of nutrients, back to simple, straightforward processes like settling just in the water column of solids coming out. So a lot of things going on that are interrelated. So what is the project? Kind of an intro background to it. About, say, around 9 or 10 years ago, Wes approached a gentleman that owned this piece of land nestled between the Clackamas River and Carli Creek that received a lot of stormwater runoff and had been getting hammered for decades, the creek. And the gentleman that owned the farm was named Mr. Carli. And when Wes approached them, he asked if he would like to sell his land, because he was getting a bit older, and Wes would take the land and care for it and eventually build some sort of treatment system on it. It wasn't quite clear at the time, but a couple of years after the purchase went through, planning began, and the water quality project with these 3 goals were kind of fleshed out. As planning was going on, it took, I think, let's say like 2 or 3 years to do the building. And in the fall of 2018, the $3.5 million project was finished and received, its first drops of stormwater from the 400-acre catchment upstream. The catchment's extremely impervious, a lot of hard surfaces, rooftops, parking lots, roads, and about 70,000 native plants went into the planting and installation. So it was a lot of brand new plants just getting established. And then 83 large wood and beaver analog structures were installed on Carli Creek itself for habitat reasons and water quality treatment reasons, as well as some beaver analog structures to try to promote and invite some friendly beavers into the area. So what were the project features, kind of in a bird's eye view? I want us to mainly focus on the white dashed line that shows the flow of water from the beginning, which is a stormwater outfall from the catchment, in the middle right of this image, through the different treatment units, and then eventually to Carli Creek before commingling and meeting the Clackamas River, just kind of off screen to the left of this image. So the water, like I said, starts by falling out of an outfall, going down about 6 or 7 step pools, which help decrease the water's elevation and dissipate energy. Then the stormwater enters this retention pond that slows down the flow of water even further, helps settle out any solids that are in it, begins contact with a bunch of vegetation. It flows under this maintenance road through a flow control device that sort of helps meter the water into the bioretention cells while still providing a lot of retention in the pond itself. This kind of mitigates any like huge storm flows, really does a good job of that. Then the water flows through these bioretention cells. I show it as one here, but there's kind of 3 separate ones with some defined kind of transition points. And then finally flows off of what I call the treatment terrace, where it's been for the whole time, into this backwater channel that is hydrologically connected to Carli Creek. It's intended to flood during heavy rainfall or when the Clackamas River is really high, which it might be right now after the rain we've had, and just kind of, again, dampen out storm flows and allow time for water to just sit there and provide a refuge for any migrating fish as well. So here's a conceptual model to kind of think of how pollution is conveyed from a catchment into the constructed wetland. So I'll just walk through it here. In the top right, we're looking at precipitation falling onto either one of 2 kinds of surfaces. It can land on pervious surfaces and infiltrate into the ground, or onto impervious surfaces and pick up different kinds of pollutants. Here I show as 4 kind of categories: solids, metals, nutrients like nitrogen and phosphorus, or bacteria like E. coli. And when that precipitation picks up those pollutants, I, in my mind, call it stormwater, and then the stormwater runs off into our MS4, or the separate storm sewer system. It can also be called, which is what I was introducing before. Once it's in the MS4, it is then conveyed to either the constructed wetland or to the headwaters of Carli Creek, which was partially buried in the development of the industrial area. And the headwaters are really just stormwater outfalls now. So the water is kind of diverted away from the creek. So not all of the runoff goes directly to the creek anymore. Some of it gets treated. And then ultimately to the Clackamas River. So why care about this system? Why care about constructed wetlands at all? This constructed wetland, like I've kind of alluded to before, is pretty young. This project happened in the 3rd winter, so it only had 2 growing seasons before I started my study. It's a good. This data will give Wes a good idea of a baseline of water quality around the wetland and on the creek, and can help inform future studies that might ask different questions. Regulatory compliance. Wes owns an MS4 permit from the state of Oregon, and one of the monitoring objectives in that permit is to assess BMP effectiveness. And constructed wetlands are a form of a BMP, or a stormwater control measure, whatever term you want to use. And then another reason is adaptive management. This constructed wetland is small relative to the catchment. There's for every 1 acre of constructed wetland project area, there's about 243 acres of catchment. So anything going on in the catchment is going to impact this wetland intensely and quickly. So keeping an eye on how it works and if it's working and how well it's working is an opportunity to change how we manage or maintain the wetland if we start seeing things we don't want to necessarily see from a management perspective. And last are beneficial use protections. This functioning wetland can help act as source water protection because there's several drinking water intakes on the Clackamas River just downstream of where Carli Creek combines with the Clackamas River. There's also listed species, salmonid species that use Carli Creek as like a refuge as they migrate up the Clackamas River. We found them in surveys and such, so want to keep this thing protected and functioning. What were my research questions for this project? One was, how well does the wetland reduce pollutants that I measured on a concentration and mass basis? And I'll just explain what those are in a second here. And then are there any environmental variables like weather-related variables or constructed wetland specific variables that could explain treatment effectiveness on the terrace? Any varying treatment effectiveness on the terrace. So I kind of looked at that too, and I'll get into that real quick here. So what was my study design to answer these questions in order to balance, resources and the time I had to do the monitoring? I chose 5 sites on the project and monitored 14 different events, the combination of composite samples, grab samples, and some continuous stations at certain sites. My first site was just at the stormwater outfall above the step pools. My second monitoring site was between the retention cells and the retention pond. The last site was kind of the outlet of the treatment terrace. I thought of it as. So I did a lot of comparisons of treatment effectiveness between my monitoring point 1 and monitoring point 3. Then I had 2 sites on the creek, upstream and downstream of the constructed wetlands influence. I also measured precipitation using a gauge Wes maintains at Rowell Middle School in Milwaukee, and looked a bit at flow data on the Clackamas River using the USGS gauge in Oregon City. So here's some photos of some of the installations we did. At site 1, because it was piped, we just installed a submerged AV sensor and a flow meter to get a sense of the flow rate and storm responses, which were extreme as you could expect with all the imperviousness of the catchment, and everything was just being piped right to the creek and the wetland. And then at site 3 and 4, we used a stage discharge flow measuring approach because it was just an open natural channel, which meant we needed to install these staff gauges and do some discharge measurements and create a reading curve. The parameters I monitored are listed here. I did a bunch of field measurements with hand meters. I measured hardness, some solids, E. coli using a grab E. coli alert method, a couple different nutrients, and several metals in total and dissolved form. And this photo just shows site 3 and with a couple of our composite samplers hanging out, chilling there next to my my data logger, and the tubing going into the creek to collect samples. Lots of fun, lots of driving that van out to the site by myself. And so when I say like water quality treatment effectiveness, what metrics did I use to determine whether it was effective or not. And these kind of related to my research questions. So I looked at concentration percent reduction, mass percent reduction, and log reduction. Log reduction I just used for bacteria because bacteria results are typically orders of magnitude different. It just kind of helps it simplify the difference between two values when your results and your data are that largely apart, that different, I guess. And concentration percent reduction, the easiest way to think about that is whatever your result for, for example, copper is coming into the terrace, and then what is it going out and the percent difference there. So a positive reduction, a positive number would indicate that the copper was reduced on the wetland. A negative reduction number would indicate that copper increased from the inlet to the outlet of the terrace. The mass calculation is just like that, but rather than using concentrations only, we incorporate a volume component to the mass number. So this accounts for the volume change that might happen on the terrace, which we did find occurred a lot, and is, I think, an important treatment process on the terrace. So we wanted to measure that too. I also looked at how my data compared to Oregon water quality criteria. There were some criteria for certain parameters I measured, like metals, for example, copper. There's criteria for bacteria, and I also looked at this international stormwater BMP database, which some folks may be aware of, lists treatment effectiveness for different BMPs, like bioretention cells, across a bunch of studies. They do a statistical analysis of the results and publish some freely available, publicly accessible results. And so I kind of looked at that too. Both of these just to benchmark what my data looked like compared to some other relevant standards or benchmarks. Here's a map of just where my project site was in relation to the Willamette River and the Clackamas River, where the Rao Middle School rain gauge was, and a couple other City of Portland hydro rain gauges nearby, as well as the USGS flow station down in Oregon City, just to kind of give a sense of it. So first graph I'm going to show, a lot of my graphs are, my figures are kind of in this time series format. This shows that this is the Rao Middle School rain gauge data during my study period, which is about mid-October to mid-April. And this shows the daily precipitation results in the black line, and the 14 rain events that I conducted and kind of where they fell between big rain events. And then the red and blue lines are just daily and monthly averages from the record. But there were some pretty big rain events during my study period. There's also, if y'all recall, 2 years ago in February, that pretty catastrophic ice storm that hit our area, affected a lot of different utilities, ours included, really hurt the treatment plants, and our guys did an awesome job getting through that challenge, but that was all during the study period. So first research question: how does the wetland reduce pollutants on a concentration and mass basis? And this is sort of like how I started the project, having fun in the field, collecting samples, running around, bright-eyed, and then I have hundreds of data points to sift through and try to make sense of and answer these questions. And that's sort of how I looked at the end of it. So before we get started, I want to get everyone orientated on what the figures I'm going to show you, how they're structured. So there'll be several panels. Each column is one of the 5 sites that I monitored in the data over the study period. and each row will be a different pollutant, whether it is total suspended solids or E. coli or something like that. And on certain slides, I have some benchmark lines on specific sites or across all the sites, just showing some statistical summaries of kind of what the data look like across the entire study versus these other benchmarks. So first set of results I want to show you, if you'll let me use a skiing or snowboarding analogy for the rest of this, we're going to go on the bunny slopes and start easy, and then we'll get into more complicated stuff. So first off, bunny slopes, a bacteria. The main takeaway from the bacteria results I got were that the treatment terrace removed bacteria really well. Not in every single event. There were a few times when my bacteria results increased from between site 1 and site 3, which is the treatment terrace, these 2 points. I don't think you can see my mouse cursor, but and then site 4 and site 5, if you remember, are the upstream on the Carli Creek above the confluence with the constructed wetland backwater channel, and then downstream is below it. So on those 2 sites, I have that red Oregon water quality criteria single sample line, just showing how almost all the results didn't really change that much above and below either, but were usually below that single sample criteria. But you'll, as you kind of notice on this log scale, the Y is on a log scale for E. coli, that there are wildly variable E. coli results in the influent, and then we saw a lot more less variable, kind of lower general results. And the log reductions we saw here ranged from -1.3, which would mean that increase I was talking about, up to 3.0, which is something like 99.9% or something reduction in some events. So pretty good. And then on the creek, the same thing. A couple times bacteria went up, comparing the upstream and downstream sites, and most of the time it was reduced. So what were the sort of mechanisms that could explain these bacteria being decreased, and how do they compare to some literature I read about other constructed wetlands and their bacteria changes and treatment? Well, when bacteria are floating through a water column, there's a lot of different ways they can be adhered or inactivated or destroyed. There's biotic ways, like having predator bacteria in the water column, that when bacteria might, or when like E. coli land on a stalk of rush or sedge or something in the water, in the constructed wetland, these predators will come and destroy them. There's also natural sunlight that could happen. If there's enough time and the bacteria are getting enough of it, it could be destroyed. They could bind to particles and move between the sediment, kind of the sediment, I don't know, or either between the dirt or the soil and the water, back and forth. And so my results and the log reductions during this study were generally in line with some other studies of constructed wetlands in Australia and Texas, showing around like 80% or so E. coli reduction across the constructed wetland. So next we're going to go to it's green circle, and that's solids. A little bit more complicated. Here we have TSS, total dissolved solids, and then total solids on the bottom. And one of the biggest takeaways is that a lot of the solids entering this constructed wetland are in the dissolved fractions. So colloids, things smaller than 0.45 microns, salts, that kind of stuff, which is useful to know if you're thinking about the types of mechanisms that will, potentially remove solids associated pollutants like certain metals or bacteria. And also that there's not a lot of TSS entering the system. The Y scales are different on each of these pollutants, so that is important to disclose. With the exception of that final April event where there's a lot of TSS leaving the treatment terrace and not much coming in. I think I found that most of the solids in the system are TDS related. And yeah, we'll talk about concentration and mass-based reductions next. So this graph attempts to get at using the daily precipitation figure from before and overlaying that in time series with concentration and mass-based reductions for each event. So for example, on the top row, we're seeing positive bars for total solids and total dissolved solids on a concentration-based reduction, which means that between the beginning of the terrace and the end of the terrace, we saw total solids being reduced. And we can look down at daily precipitation and see, yeah, there wasn't much rain happening before that. So not really analytical or quantitative in the sense of comparing reductions in rain, but just qualitatively looking, seeing if there's a pattern there. And so a lot of these negative bars are on the total suspended solids pollutant. So that to me says that on a couple events that we saw TSS being exported from the system. But if we recall the graph before, there really wasn't much TSS in there to begin with. So it's probably changes in small numbers being reflected as huge percentages. So in concentration, there's sort of like, I see like mostly positive results, which means retention in the wetland. But when we get down to mass, it's a lot more pronounced, I would say. The reductions are typically higher, regardless of the time of year during the steady period for all of them, except for TSS and TS in a couple instances. And that is something we'll talk about a little bit later. Why are the mass reductions higher typically than concentration-based reductions? I gave a hint earlier. I'm sure you guys already know the answer of it, but what might explain why TSS is never really even entering this system from a really industrialized catchment? Well, the main reason is this ginormous technical term, hydrodynamic separator that was installed in the project upstream of the constructed wetland. It's like one of the biggest they make, but how it works is it captures solids and floatables like oils and trash in the stormwater before it's discharged into the treatment wetland. So this is kind of a great example of gray infrastructure doing its part in minimizing solids getting to the constructed wetland and then later needing to be taken out through dredging or something. So this is just a piece of equipment that gets regularly maintained by our field crew and has the solids removed periodically. So onto the green circle, blue square, medium difficulty, are nutrients. So here are my nutrients results. Ammonia, I measured nitrate, nitrite, oxidized nitrogen, and then total phosphorus. Here in these figures, I did keep ammonia and nitrate, nitrite on the same Y scale. And just to show how little ammonia was in the system, kind of similar to TSS from before. And then nitrate, nitrite, how that compared to it. And then just some of those, I didn't say this before, but these green bars in the middle column are those international BMP statistics for bioretention cells, just showing how my data compares to others. And something notable here is this thing, yeah, that my nitrate data leaving the bioretention cells is generally a lot higher than the median nitrate from other bioretention cells in this BMP database. Total phosphorus, it wasn't really an interesting story there. There was that high spike in late April again of total phosphorus, and that probably coincides with that high TSS result I got, which my best guess is it was just poor strainer placement on my part. I probably just put the strainer next to too low on my sampling, the sampling location, and might have sucked up some sediment or too much algae or something, because nothing else I could think of really made sense for that. But what about concentration and mass-based reductions? Well, nitrite was pretty much reduced throughout this entire study period. That's the green bars on these figures, by concentration and a mass basis. But ammonia shows a lot of export. A lot of these negative bars on here indicate ammonia being exported from the treatment terrace between sites 1 and site 3. And I think that might again be a consequence of really low ammonia values to begin with, but I don't think it's worth just brushing over. There's something there, and I didn't really kind of get to the bottom of it. And during this study could be, biological breakdown, or an actually ammonia being generated somehow in the constructed wetland. But yeah, so that's the data here. It seems like these rain events kind of had some like pulse effect, particularly in like over the new year. I don't know if folks remember that December either, but the river was extremely high. The Clackamas River had really high stage during the couple of these January events, which may have impacted some of the processes happening up on the terrace. My best guess is that time. I got a little bit of time. This isn't something my study evaluated, this nitrogen cycle and all these different possible species and mechanisms going on, but I just show this figure to kind of exemplify how many different nitrogen species there are to begin with, and how when you involve plants and their root systems and the bacteria associated with their root systems, how complicated it can get very quick. So a little more useful is looking at other constructed wetland studies and how they reported nitrogen and phosphorus reductions or mass removals. Unfortunately, a lot of them usually only report total nitrogen and total phosphorus. They don't speciate beyond that, but there were a few, and nitrite was definitely more in line with the literature in terms of how much it was reduced. But again, this constructed wetland is very young, and there may be some story that's important to tell about how the land that this constructed wetland was built on used to be farmland and was most likely fertilized for decades. So there may be some native fertilization effect that might be able to explain some of this nitrogen showing up. Okay, last but not least, we got metals here, showing zinc, copper, and lead, because cadmium there wasn't much going on with cadmium and wasn't much happening with mercury. So I looked at these three metals, takeaway here is that for nearly all of my events, zinc, copper, and lead results were always below the Oregon water quality criteria on the creeks. And that with the exception of lead, most of the fractions of zinc and copper were dissolved. You can see these blue lines are the dissolved fractions versus the total fractions, and how they were a lot closer to the total lines compared to lead, where there's almost never any dissolved lead. It's mostly all solid total associated lead. So here, this is concentration-based reductions for these different metal species, comparing them to daily precipitation. This shows, to me, the most interesting story that kind of matches with the mass-based reductions is this February event when we had that big ice storm. It just seems to like kind of reset and shock everything into this new normal. When we get a lot of export of nearly all of the metals and all of the fractions, with the exception of zinc, somehow always being pretty well retained, probably because it's maybe solids associated more so than the other metals. But and so again, this is the mass-based reductions. As before, mass-based reductions are typically more positive across all the metals, except for when that February ice storm hit. It just kind of threw everything out of whack. Might have just like shocked a lot of the plant and bacterial communities in that wetland, enough to take a couple of weeks for things to get back to normal and begin retaining metals again. So it's just an interesting shock that showed up in the data. So here, show on the right, just a lot of different mechanisms involved with retaining metals. Didn't really get into elucidating or figuring out which one was dominant and which one wasn't. But the literature showed that for this study that occurred in Sweden, which is not exactly the same climate as Portland, Oregon, or Clackamas County, they had really, really good metal removal compared to this Carleton study. My data tracks more closely to the Carleton study, which was in the United States. And then just some general rules from the literature: lower ORP and flows, less extreme storm flows, higher pH in organic matter, which probably takes a while to build up in a constructed wetland, usually means retention and hanging on to these metals a little bit stronger, versus higher flows and poorer density, macrophyte and plant density likely means scouring and export of different metals. So why are mass reductions consistently larger than concentration-based reductions? All about the volume. This zoom in is just about a 3-month time period during my study, comparing the flow monitoring I did at 0.1 and 0.3. So the piped part above the treatment terrace, and then the outlet of the treatment terrace. And the outlet trace is below the inlet trace a lot, which means a lot of infiltrations happening on the terrace, a lot of water soaking into the ground. And that's reducing the volume and showing up as higher mass-based reductions. So analysis of all the storms in this window you're looking at showed about a 36% peak flow reduction. So every 100 gallons that comes into the system, only about 64 gallons are leaving it, which is an important part of why they designed this constructed wetland. There are several limitations of my study. There's the accuracy of my peak discharge rates. There was a couple times where I probably could have improved that. There was definitely a lag, like shown in this figure on event 12, from the time the peak of a stormwater event or precipitation event hits monitoring point 1 and the time that peaks and leaves. That's an example of delayed peak. I did not account for that holding time, that retention time in my calculations. I just used the 24-hour composite to measure how much volume went into the reduction numbers, the reduction formulas. The precipitation record was really short when I was comparing that with the reductions. I did not target storm events, which is probably when a lot of pollution is being delivered. Should be interesting to do in another study. The wetland is young, and I did not study anything about plants or other potential non-water quality benefits to the constructed wetland, which might be meaningful to some folks. So takeaways: mass-based reductions do occur on the terrace, but sometimes it acts as a sink for like nitrate, keeping it in the system, and other times as a source, such as letting lead leave, some low-level total lead. The flow reduction is an important mechanism. Understanding nitrogen dynamics is difficult. I did not measure all the species involved, but the data suggests that there's denitrification or uptake happening even during the rainiest season. But there is some more to learn about ammonia. Total dissolved solids reduction in the system suggests there's some absorption or flocculation or precipitation happening, and that ongoing maintenance is going to matter. If you want to, it's important to maintain vegetation and potentially even to the point of harvesting it occasionally and removing it so phosphorus doesn't leach from the harvested vegetation. Managing the soils and the retention cells is important to maintain volume capacity, and just high density vegetation will help continue increasing the surface area available for, active sites and metals to be kept in the system. These are some of my future study questions. What about the plants? What about hydraulic loading? What are the actual mechanisms on the terrace? How could we create a study to understand that? Is there more fine level spatial detail that would help explain some of that versus just looking at one point before the terrace and after? And what about the growing season? How well does this thing perform when it's not raining and all the plants are just going gangbusters and growing as fast as they can in the summer? Couple acknowledgements. Couldn't have done this without these people here in these groups, I swear. The water quality lab at Wes, Eric and everybody taking all my samples and analyzing them for me and getting me the data. My graduate committee and the lab group that had to listen to this presentation 1,000 times before and in 1,000 times worse condition than you guys see it today. Some funding from a scholarship, and of course, my family and friends and wife for supporting me. Thank you for listening, and I'll take any questions. I can pull that slide back up if anyone wants to see something one more time too. I might be out of time.
You got 5 more minutes. If anyone has any questions that doesn't have access to a mic, feel free to add them into the chat and I can read them out loud for you. Okay, it looks like we have a question from Mark, and he is wondering if the project area is open to the public.
Right now, the only publicly accessible route is via the Clackamas River, unfortunately. So you'd have to float. You'd have to take a float or a kayak boat to get to it. But I'd be more than happy, and a lot of my coworkers would be happy to give anyone a tour if they'd reach out to me. I could take you guys down there and show you everywhere. It's really nice.
Looks like another question from Kim. How often does the pretreatment need to be emptied?
I think we're probably targeting to do it, maybe twice a year right now, but it also gets serviced in the event of like a fire response in the catchment upstream. It's really nice for that in that sense, and that if there was like a massive oil spill or an accident and fire department had to respond, all of that wastewater, all that stormwater would hit that separator first, and then our guys could go kind of head it off before it hits the wetland and then the creek after that. So it kind of serves as like another layer of protection. And our guys, I think, intend to monitor that separator when that happens. Thanks everyone for listening.
Thank you, Chris, for the presentation. Just so everyone knows, maybe like a minute or 30 seconds before the session ends, there should be a notification that pops up to let you know that the session is ending, and it should automatically shoot you back out to where you can choose the tracks again.